Hello, everyone. Thank you for joining us this evening at this hour just before weekend. It is also noon in Minnesota right now. Thank you, Professor Uğurbil, for accepting to give a lecture to us today. We have the great pleasure to welcome Professor Dr. Kamil Uğurbil to discuss multidisciplinary studies in neuroscience and FMRI, and also his career pathway in this domain. My name is Banu Erdem. I am a second-year PhD student in neuroscience with a computer engineering background. I would like to present first our club KUSIGN, Koç University Student Interest Group in Neurology and Neurological Sciences. KUSIGN was established in late 2019 under the leadership of a group of voluntary students as the first official club of the American Academy of Neurology in Turkey. KUSIGN aims to support interdisciplinary dialogue and research projects by bringing students from all faculties with an interest in neuroscience. Now I would like to leave the words to Eray, who will explain to you Career Events Committee of KUSIGN. Thank you.
Hi everyone, I'm Eray. I'm second year medical student. The main motivation of our career committee is to bring graduated students, grad and undergrad students together so that experiences can be shared, diverse career pathways in neuroscience can be seen, and also these pathways can inspire undergrad students to study neuroscience. In this career committee, we have granted undergrad students from medical or non-medical faculties such as engineering, psychology, molecular biology, and normally we want to arrange a hybrid meeting including both face-to-face and online sessions at the beginning of the spring semester, but due to earthquakes happening in southeastern Turkey, we couldn't achieve that goal. Instead, we organized this webinar. So now I want to finish my words so that we can pass to Sami Yiğit.
Okay, thank you, Eray. Hello everyone, my name is Sami Yiğit Demir, and I'm a prep medical school student. Today we are going to talk about neuroscience and Kamil Uğurbil's career. Welcome hocam. I would like to thank you for accepting our invitation and join our seminar. If you let me, I would like to summarize your resume. Professor Kamil Uğurbil was born in Izmir, Tire. He studied in Bornova Anatolian High School until his last year. After that, he applied for the scholarship of Robert College in Istanbul. After his graduation from Robert College, he was eligible to continue his PhD in USA. After completing his bachelor and PhD degrees in physics and chemical physics at Columbia University, he joined University of Minnesota in 1982, where his research in magnetic resonance led to the evolution of his laboratory into an interdepartmental and interdisciplinary research center, the CMRR. He was one of the two pioneers of the Human Connectome project and one of the 14 members of the first Brain Initiative Working Group. He was recognized by several awards and honors, including the IEEE Medal for Innovations in Healthcare Technology and two honorary doctorates. He also won Webby Koç Award in 2016. Also, he is currently the director of the Center for Magnetic Resonance Research, and he is a professor of radiology, neuroscience, and medicine at the University of Minnesota. Thank you for coming, hocam. We are thrilled to listen to you. The stage is yours.
Okay, thank you very much for the invitation. And hold on one second. But you have to enable screen sharing so that I can share my screen to show my slides. The screen shared is not enabled right now. Okay, let's see. So do you see my slide here that says CMRR, Center for Magnetic Resonance Research?
Yes, we can.
Okay, great. All right, so let's start. Well, again, thank you for the invitation and for the introduction. I'm happy to join you and give you this lecture. It's a slightly different lecture than I usually give because of the fact that Sami asked me if I would actually talk a little bit about my career, how did this, or what he said was something like the decisions you have made that affected your career and how it has evolved over the years. So, I will actually do something like that and tell you a little bit about my background, where I came from, what I think were important points in my career, and maybe some lessons I learned from my career about how to actually develop a career in science. Also, I'll give you some description of the laboratory. I won't be specifically talking about a particular neuroscience problem in great detail as a result of the fact that this is somewhat of a general lecture. It is a little bit more about some of the philosophies that we have pursued in developing this current laboratory that I lead, which is called the Center for Magnetic Resonance Research. So, as Sami has mentioned, I was born in Tire, which is a kasaba. It's a town, a small town near Izmir. It's not such a small town anymore, actually. It may have a population of something like 100,000 at the present time, but when I was growing up, it was really a very small town. It was something like 20,000 people, very agricultural town. And you could still see people in Tire, coming to the bazaar, for example, on horseback. Horse carriages were commonly used, as well as tractors, and some, private cars were very few, etc. It was a town very far away from science and very far away from research. I mean, Turkey, when I was a kid, was very far away from science and research as well. But I had great teachers in my life in Tire. Let's see. So this is a picture of me graduating from the elementary school in Tire, and right next to me is my mother and my grandmother. And my mother was one of my greatest teachers. I had to every morning, get up and go through my homework with her before I went to school. And this is a picture of my elementary school teacher and her three most successful students. And she wanted to take this picture when we graduated. And maybe the seeds for science was really installed in me by this teacher who, at the time of the graduation, gave me a book. It was a small red book that said the title of the book was 12 Life of like 12 Scientists. And she wrote in the book that I hope you will be the 13th. And so I think at a very early age, I had this idea, maybe science may be a career, but of course, I had really, when I said I had this idea, it was a very vague idea, or it was a fascination with science she introduced in me. And I went to, that was the 12 Bilim Adamı, that was the name of the book, and she wrote, Ümit ederim 13. sen olursun. So I went to a great school after that for middle school. I mean, really fantastic. This is, at the time, it was called İzmir Koleji or Bornova Marif Koleji. It's this, what is now is an Anadolu Lisesi. But it was very different at that time. This was my class, 25 students. And, English and Turkish education. And this enabled this school. I really had a fantastic experience there. And many of my classmates have actually developed their life and had made major contributions to Turkey, including foreign ministers, industrialists, etc. I went from İzmir Koleji to Robert College. This is one class picture taken in Robert College in the Yatakhane, if you like. At that time, I think this was like the last year, Lise 3, if you like. And there are also many people from my class who made major contributions to Turkey as well as to science. For example, the person sitting in front of me here is Professor Nihat Berker. He is now in Turkey, and he was a professor at MIT. He is now a professor emeritus at MIT. Very accomplished, famous theoretical physicist who brought, if you like, theoretical physics to Turkey. So, very productive group of people. But what was so wonderful about Robert College was the fact that in those days, I mean, of course, today a lot of people come to the United States to go to college, but in those days, this was very rare, except Robert College had some connection to the United States, and usually, few people, 4 or 5 people would go to the United States for university education. Nihat Berker was one of them. He went to MIT as a student, and I was another, and I went to Columbia University for my undergraduate study. And there's a picture of Columbia University's famous campus in the middle of Manhattan. This is so-called Law Library that appears in many places. It actually appears in movies. It's actually a famous location. And when I was in Columbia University, I didn't really know exactly what I really wanted to do. But I took an electricity and magnetism course. It was taught by a wonderful professor, and used this book by Edward Purcell, Electricity and Magnetism, and I fell in love with physics. It was really a great experience. I decided to then major in physics. So this is a picture of me in front of the physics building, so Pupin Hall at Columbia University. It's a very famous building. It's actually a national monument now, which is good and bad in the sense that you cannot do much to this building anymore. This was a place where quite a lot of work was done in development of the Manhattan Project, which then in fact was involved in the development ultimately of the atom bomb, but many other very remarkable discoveries were made in this building. So, in fact, this is one of them. Isidore Rabi was a professor at the physics department. When I was the student there, he was actually a professor emeritus, so he was not an active professor, but in that particular building, the discovery of what is today nuclear magnetic resonance was done, and for which he got the Nobel Prize in 1994. I mean, this is kind of a very different kind of a instrument, if you like, from what we do today with magnetic resonance. But nevertheless, this was the first work that demonstrated that nuclear spins can flip and can have resonance. And as I mentioned, he received the Nobel Prize for it. So I was in an extremely rich intellectual environment at Columbia University, and exposed to many fantastic scientists. I mean, when I was a student there, actually, there were 3 Nobel Prize winners who were already resident professors teaching undergraduate classes at Columbia. From Columbia, excuse me one second, I went to Bell Laboratories. That's also an extremely famous place, and it's an interesting, I will tell you a little bit more why I went to Bell Lab, but it was an important decision in my life, and a very famous place at that time. You know, Bell Labs have fantastic contributions to our life today. For example, the transistor was discovered in Bell Labs, laser was discovered in Bell Labs. And it was not just also practical things like that, but there were a lot of basic science was going in Bell Labs, and including, you know, the background radiation in the universe was detected and explained in Bell Labs, for which, people had received Nobel Prizes as well. So fantastic scientific environment. And in many ways, I owe quite a bit, of course, of what I do today to my time and Bell Labs, as I will explain later. Here's a picture of me in Bell Labs. Excuse me. You probably cannot recognize it, but the person over here with the beard is me, together with 2 of my colleagues, looking at this instrument which we used for studying cells with magnetic resonance, and it was not imaging at that time, but we used to do experiments like this. It was the first time we were trying to use the magnetic resonance phenomena to detect signals from inside intact cellular organisms. up until then, magnetic resonance was a tool of studying the molecules, for example, in organic chemistry, or whether you synthesize the right molecule or not, or in biochemistry, you know, solving structures of proteins, etc. That was the use of magnetic resonance. But I was actually very attracted to this work at Bell Labs, where they were trying to do something totally new, that was essentially trying to use magnetic resonance to detect signals from inside the cells. So this is a suspension of E. coli cells, and you have an NMR spectrum from it, and a phosphorus NMR spectrum. And what you see are signals that come from phosphorylated metabolites, and they are very important metabolites in the life of a cell, like ATP. But in bacteria, actually, it is it's not just ATP. It is so it's labeled NTP, nucleotide triphosphates, you know, PI, etc. And this was a totally new world, a new endeavor, and this is what attracted me, and that is why I ended up essentially. I had several opportunities to go to different laboratories, but I went to this laboratory because I thought, wow, this is a new thing, and I think that this has a potential in the future. And I grew with that, with this particular effort, because of the fact that it was new and it did explode, and it did become today not just looking at bacteria but human brain. It has, but the origins are right here. Now, I want to show you a little bit about the environment that I was in, scientific environment I was in. This is something called neuro tree, or you can actually have these trees. It's essentially kind of like a tree of people who were, you know, who you are linked to, who were your teachers, and who were their teachers, and who are your students. So if you look at me, for example, right here, it says Kamil Uğurbil, UMN, and then I worked with Bob Schulman in Bell Laboratories. I worked with Richard Burson at Columbia. He was my PhD advisor. And then all these people that come after me are all the people that worked, who worked with me, and you know, they have their own labs, or they continue work in CMRR, etc. But it's very interesting. So when I look at this, so I'm right here, and my PhD advisor, person who was heading the Bell Labs laboratory I was in, and if I look at Bob Schulman's ancestry, scientific ancestry, there are a lot of very, very prominent people, like Francis Crick, Nobel Prize in Physiology and Medicine, you know, solved the DNA structure. You know, Ernst Rutherford, another Nobel Prize. You know, Lawrence Bragg, the Bragg diffraction, another Nobel Prize. Willem Röntgen, you know, Röntgen, another Nobel Prize. So full of Nobel Prizes in the ancestry of these people that they've trained. And my PhD advisor also, okay, there's another Nobel Prize in the tree of Bob Schulman. And if I look at my PhD advisor, there's Lamb, Nobel Prize in Physics. There's Van Vleck, again, Nobel Prize Physics, etc. So you get the point that I worked with people who also worked with very famous scientists. And so in my scientific ancestry, there's quite a lot of fantastic people who made major scientific discoveries. Let's see. And then when I was looking at that, I came across this article which says Scientific Prize Network predicts who pushes boundaries of science. So indeed, in fact, this article, essentially what it says is that, you know, if in your network there are quite a lot of, you know, fantastic scientific people who have received a lot of scientific prizes, then you have a pretty good probability that you may also end up doing something good in science. Now, of course, when I went and did my PhD and went to Bell Labs, I was not aware of all of this. It was not a conscious decision, but let's say that I was really lucky to end up working with really wonderful scientists and who had made major contributions. And it's that intellectual milieu that I think has made an imprint, which probably really shaped how I approach science and what I try to do in my own career. Anyway, let's go back to Bell Labs. I'm sorry. From Bell Labs, I went to Columbia University again. Actually, this was as Sami's history of my career a little bit off in the sense, and that from Bell Labs unfortunately broke up more or less towards the end of my stay there, and many people left. It became much more of an industrial laboratory as opposed to a big, fantastic scientific laboratory. I went to Columbia University, and I was only there for a few years. Then I got an offer from University of Minnesota. Oh, sorry, University of Minnesota. Columbia tried to counter that offer, but at the end, I decided to move to University of Minnesota. And I think that's it may be an interesting information as to why I made that decision. I mean, Minnesota was willing to invest a lot of money into a research program that I wanted to develop, and Columbia tried to counter it, but it was a very complicated offer. I had to collaborate with this person, that person. I was not very happy about it. And so many may say, why move from Columbia University, such a famous university and Ivy League school, etc. But I, had some confidence in myself, and I thought, well, I hope that I will be able to, and I think I will be able to develop a wonderful research program, even in a university which is less famous than the one that I'm in, but still a very good and wonderful university. And it worked out very well in that respect. But coming back to Bell Labs, these are two of my Bell Labs colleagues. I mentioned Bob Schulman, who was heading the department at Bell Labs when I was there, and another colleague from Bell Labs, Seiji Ogawa. And it essentially, I want to also emphasize that people in science that you interact with do make enormous contributions to one's career, and they're very important. And Seiji has done something that was very intriguing to me, and he discovered this phenomena called the Bold effect. And what he noticed was, if you take a rat brain and you put it in the magnet, and there's actually a very good article that Seiji and I wrote describes this. It was an accident, but accidents favor the brains that are ready for them. And the rat died in the magnet, and then he noticed that the rat had all these the rat image. This is an MR image of the rat brain, had these dark lines, and these dark lines, it turns out, correspond to veins. And the veins contain deoxyhemoglobin, and when the rat dies, deoxyhemoglobin becomes, hemoglobin becomes deoxygenated, and deoxyhemoglobin is paramagnetic and causes these dark lines in the image. I'm sorry, I just recovered from COVID, but so my throat is not so fantastic. So this is the explanation. So you have blood vessels. In the blood vessels, you have hemoglobin, and oxyhemoglobin is diamagnetic, so its magnetic properties is like tissue. But if it loses its oxygen, which it does when the rat dies, you get deoxyhemoglobin, and the oxyhemoglobin is strongly paramagnetic, and it causes magnetic field inhomogeneities around these blood vessels. And those magnetic field inhomogeneities then, in fact, is what we are detecting in those images. Now, there was another discovery a long time ago that was very important also in the development of functional imaging. And this was this development by Roy and Sherrington, who said that blood flow in the brain is coupled to neuronal activity. So Seiji and I were thinking that, well, okay, if you have increased neuronal activity, then you have increased regional blood flow, and then this should lead to lower deoxyhemoglobin content per unit volume in the brain, because oxygen consumption rate does not increase commensurately. That was also known from other studies in the brain at the time. So this idea, this sort of observation, was in fact the basis of why we decided, Seiji and I together, to try working on trying to use this effect, this Bold effect, to generate functional images in the brain. And at the time, I was waiting for a 4 Tesla magnet. This is a very it looks like a very primitive instrument, but this is a 4 Tesla magnet. At the time, it's a very advanced magnet. Again, what you are looking at is actually the doer. In essentially, the inside of this magnet is at 4 degrees Kelvin and superconducting magnet, etc. Very advanced of that magnet of the time. And when this magnet arrived in Minnesota, we actually performed the first functional images there. And this is from our publication showing that, there's a subject in the magnet, and we turned lights on, the subject is in the dark, we turn lights on, lights off, and we see that in some parts of the brain, of course, the signal is tracking lights on and lights off, and in other parts of the brain, nothing is happening with the lights switching off. And if you look at the difference then between periods when you have lights on versus lights off, you get an image like this, and this is actually an image of the visual cortex of the brain responding to the lights. And this started functional imaging. There was another work that came at the same time from another laboratory, and these two papers essentially became the foundation of functional imaging, which is the primary technique that is used today to study human brain. Now, why can we do that in the human brain? Because we really don't have single neuron resolution. This is not like electrophysiology where you stick an electrode and you get activity from a single neuron. But we don't necessarily need to activity of each single neuron, because in the human brain and in many brains of highly developed species, even like cats, rats, monkeys, etc., brain function is spatially organized. In other words, there's a functional map, and that means that many neurons that actually do very similar things are grouped together. So we don't have to see individual neurons, although actually there's of course a reason why it would be good to see those, but nevertheless in a functional map, clusters of neurons can display similar activity, and when you have such clusters, we can essentially map those ensembles of neuron using techniques like magnetic resonance imaging. And this was first shown by Broca, and you may know about Broca, but this was the first demonstration. And Broca had a patient, and this patient had suffered a stroke, and the patient could not talk actually, but otherwise he was normal. And Broca followed this patient, and subsequently, when the patient died, he did an autopsy, and he showed that there's a region that's sort of like very dark over here where this patient had a stroke. And this area is now called Broca's area, and Broca's area is of course involved in speech. And with functional imaging, of course, we are very much able to map Broca's area. It's right over here. This is a subject doing silent word generation inside the magnet. Of course, there are more areas than Broca's area involved in this particular process, but of course Broca's area is showing up in this map very easily. Today you can do a lot of things with functional imaging. For example, one of the things that people have done is study the human visual system extensively. We know that human visual system is retinotopically organized, and we know that it classifies objects, like, if you're looking at a fruit, or it classifies space, etc. And all of these properties of the visual system, which were to some extent known from macaque studies, but they have been actually expanded and extensively studied in the human brain for the first time because of functional imaging. Now, we have a philosophy here in this laboratory, and essentially philosophy is one that subscribes to this thought coming from Freeman Dyson, who wrote in one of his lectures that he said new directions in science are launched by new tools much more often than new concepts. And this is not unique to Freeman Dyson. Of course, in the brain, Santiago Ramón y Cajal, who actually gave these wonderful drawings of neurons, was able to do so not only because he was a very good artist, he could draw these things, he was a great scientist, but at the same time he had a new tool that was not present before, and that was the microscope. And you can see the Ramón y Cajal sitting in his laboratory with the microscope looking at these brain slices. So new tools are really important for new discoveries. And here's another example. For example, I had the pleasure to chat with Leroy Hood about a year ago, and Leroy Hood is a very famous name in the DNA sequencing, which led to the Human Genome Project. And he credits his advisors who told him, if you want to practice biology, do it on the leading edge. And if you want to be on the leading edge, invent new tools for deciphering biological information. And this is his advisor, William Dreyer, and Leroy Hood had said that Lee Hood was indoctrinated by me into this importance of instrumentation and methodology when our theories clearly were way beyond what we could do experiments for. Anyway, we subscribe to this philosophy that tools are extremely important, and so we combine essentially a philosophy in which we undertake tool development, which fits also to our background, my background in physics, and then we try to apply those tools. And for us, the tools have been magnetic fields, and so we started with a 4 Tesla at a time when clinical magnetic fields were 1.5 Tesla at lower, and then we went to 7 Tesla, and then we are now working at 10.5 Tesla. And in many ways, we cannot build these magnets. We were lucky to have a partner in a company that can build those magnets. The rest of the equipment usually we put together ourselves, but these days you can buy some of these equipment now commercially. And then we had to learn and develop a lot of new physics to understand how you image human body at these very high magnetic fields. However, because of this focus in tool development, we have now in our laboratory many magnets. We started with animal magnets, then went to human magnets. So if you look at our building, you will see signs like that pointing to directions to where you can go to where these magnets are located. We even have 2 magnets right now which are in our courtyard. They are garden statues, if you like. And these magnets we don't use anymore, but they are, historically very important magnets. The silver-colored one or the steel-colored one is the magnet where we did the first functional imaging, and the white magnet is the very first 7 Tesla in the world. And 7 Tesla today, as I had mentioned, is a commercially available system, maybe the most advanced system now, that can be routinely used and commercially available for brain studies. But there was a scientific, of course, rationale for why we wanted to go to these high fields. So we didn't just say, oh, let's just go to high fields. And the rationale comes from the fact that in the brain, the spatial organization of neurons, to finally generate from single neurons to behavior is rather complex. So if you look at, and it covers a very large spatial scale. So if you look at the visual cortex, which is right here, human visual cortex, it has some organizations which are in the supramillimeter domain. But, for example, if you are looking at a view like this, the, periphery of the view maps over here, central areas map to the fovea in the visual cortex. But now if you look at a very small section of that visual cortex, there's an organization of neurons that respond to orientations. One eye versus another's eye, the neurons are segregated, etc. So there's a submillimeter or mesoscopic scale organization. So when functional imaging came into existence, MR functional imaging, there was another development, a parallel development that was very intriguing and that has caught my attention. And that was actually a paper on optical imaging. So this is an invasive experiment. It's using so-called intrinsic signals. They are looking at optical imaging, and of course they have a window into the brain. They are looking at recording from the surface of the brain with light, and they were looking at visual stimulation, and they were able to map these so-called orientation selective cells that I mentioned to you here, or orientation tuned cells. They were able to map that. This was those cells were already known. They were discovered with electrophysiology by Huber and Wiesel. Hubel and Wiesel had gotten a Nobel Prize for it, but Hubel and Wiesel, doing electrophysiology, did not know how those cells were organized. This optical imaging paper, in fact, showed how those cells are organized in the brain. And it is a very interesting paper to me. It always fascinated me how nature has solved the problem of like 360-degree orientation representation. It was a really interesting, both from the scientific point of view and also from a tool point of view, that actually these optical imaging based on hemoglobin, these are so-called intrinsic signals, that they could map it. There was another work, a really fantastic work by a colleague from University of Minnesota, Apostolos Georgopoulos, that also attracted my attention. And he was looking at using electrophysiology, tuned cells in the primary motor cortex of the brain. So he discovered that there are directional tuned cells, like these are directions, in the primary motor cortex of the brain. And then you can record from those cells and then generate something called a cell vector. In other words, this is like the line here. The direction of the line is the tuning direction of the neuron. The length of the line is actually how much a neuron fires when the monkey is going to move their arm in a particular direction. And you add all those cell vectors, you come up with a population vector, and the population vector then actually predicts how the direction of the hand movement of the monkey. That's really really interesting and fantastic work in my mind, showing how population vectors represent a mechanism in the brain. And I was thinking, well, okay, we cannot record from single cells, but if these directionally tuned cells are like the orientation tuned cells in the visual cortex, and they are clustered in some mesoscopic scale, maybe we can record from population, some groups of those, ensembles of those cells with MR, and we can do actually this kind of mechanistic work with functional magnetic resonance imaging. So the idea then became, how can we reach from whole brain studies, which we were able to do in the early days of fMRI, to this mesoscopic scale? And that became a driving force in my laboratory. And there was an idea, however, that also came from a modeling studies that we did. And the modeling studies are essentially, we're trying to understand the mechanism operative behind the functional imaging signals, and we modeled them based on vasculature. And what we noticed is that functional imaging signals really come ultimately from blood vessels, and large blood vessels, so the Bold effect associated with large blood vessels like this. This is the cortex. These are large blood vessels on top of the cortex, so-called Peel vessels. The large blood vessels, the Bold effect increases as the field, and capillaries, which is really the signals that we are interested in, they are small, but they increase as the magnetic field squared. So that actually told us magnetic fields are very important in functional imaging. So as a result, we decided to go to very high magnetic fields when, of course, no such instruments existed in the world. So we developed the 7 Tesla. This is the 7 Tesla magnet going into our laboratory. And with 7 Tesla, we in fact were able to map these orientation tuned cells and ocular dominance columns for the first time in the human brain. And it was not just for the first time in the human brain, but it was also the ability to demonstrate that fMRI can reach into those mesoscopic scale organizations. And so we did that. And we also actually, with Apostolos, mapped population vectors and showed that fMRI can indeed actually work at the level of these population vector or cell vectors. So Apostolos did experiments in the monkey, recording, and in this case, we were recording from the superior parietal lobule, and he showed that with electrophysiology, there are tuned cells. This is cell electrophysiology in the macaque, essentially showing that there are these tuned cells. I mean, the cells are tuned to different directions, but this is essentially a standardized, so we mapped everything, whether the tuning was 90 or 180, etc. We mapped it all to 0. And this is an fMRI voxel. So in one case, single cells, in one case, population of cells, but you can actually detect the same tuned cells. And you can also just, like in single cells, demonstrate that the population vector of fMRI indeed predicts direction that is encoded in those tuned cells. So those two achievements, it took us, maybe over a decade from the idea that we really want to know if we can get to those levels, and we developed instrumentation, a lot of techniques. We finally achieved all of those things, and we felt at the end, of course, like we were elated, like this mountain climber who is on top of the peak that he has reached, and we have felt this incredible satisfaction. But of course, it was a very hard job getting there, and it took a whole team of people and a lot of work to summit and make that climb. This is an example. I mean, this is now essentially mesoscopic scale functional imaging in the human brain is a very blossoming field. This is an example from our work, for example, in the auditory cortex. You can not only have primary auditory cortex, very interesting maps of tonotopy. In other words, frequency selectivity is a map in the auditory cortex. However, it's a map in the inferior colliculus. It's a map in the medial geniculate nucleus. And then even if you look at the primary auditory cortex, and actually there are organizations across the millimeter thick cortex that are important for this auditory processing. This is just one example, but there are many such examples now coming from many different laboratories. So ultra high field MR, then all these technique developments we had did indeed allow us to get there. But there are also many examples where what we achieved with 7 Tesla did not allow us to reach. I mean, we reached orientation domains, but we did not reach, organizations at subscale of the orientation domains, and there are such organizations. And ultimately, of course, we have these single neurons, and so we are still pushing to increase the ability of magnetic resonance and fMRI towards the single neuron scale. And then there's another reason why we do that, because there's another technology, namely multiphoton optical imaging, where you can detect these signal cells and they actually overlap. There's a spatial scale that is that they overlap, and the idea is that, well, you can actually go from, in principle, single neurons to whole brain networks if you combine these two technologies. So today we also invest a lot on higher magnetic fields. I mentioned also 10.5 Tesla to go into higher spatial resolution. And this is colleagues who've got the first images on 10.5 Tesla. It was a lot of work to get there. Again, it's a 1 decade long project. And there's well justified the smiles on top. We do a lot of technology development besides having a magnet. This is, for example, one of our recent detectors is so-called 128 channel detector, very complicated piece of electronics which we develop. And but we were able to then go in this direction, and we are pushing in that direction. And we are also now investing in multiphoton optical imaging as well. I have a group of people now working on multiphoton optical imaging. For example, this is from the lab of, this is the faculty member, Professor Kara, who now works with 3-photon imaging. These are all single cell activities recorded in the ferret neocortex with optical imaging. Another wonderful optical image, a phenomena which allows you to see all the tracks in the optical image and across the layers. And this is a 3-photon which reaches across the whole cortex. Really wonderful technology. Another faculty member now in our optical imaging group, for example, focuses on recording from dendritic spine. So we cover again a spatial if you like, a spatial, different spatial scales going from single neurons and to subsection of the neurons. And we can even now are working on technology where we can record these single cell activities inside the magnet. So this is work that we published some time ago now, but the technology is now just maturing. We can do single cell recordings inside the magnet and at the same time perform functional imaging using the magnet itself. So I will end there, and I want to just, of course, acknowledge my group, the Center for Magnetic Resonance Research in University of Minnesota. You know, it's a fantastic group of talented people coming from many different disciplines. It's a multidisciplinary group. You know, we have neuroscientists, computational neuroscientists, and mathematicians, and physicists and engineers. This is our engineering group, you know, which is a critical part of, as I mentioned, the philosophy of this laboratory. And also, you know, image reconstruction is a very important part. So we have mathematicians, in this case, Mehmet Akçakaya and Steen Müller. And with that, I think I stop over here, and thank you for listening.
Thank you, Kamil Hocam. We received some questions as well.
Okay.
I think that Sami will ask you order by order. Sami, would you? He had a connection problem, I think. Just a second.
Okay.
Maybe you can go and come again, Sami, or rapidly.
Maybe I can ask a question while we are waiting.
Okay.
Okay, it's very inspirational work actually on the high end of developing MRI machines. For human-computer interaction, the fMRI imaging is very important. Do you think we can have some kind of wearable fMRI that could be used for human-computer interaction anytime in the near future?
If you are, let's see, yes, in principle, yes. I mean, sort of like there are baby steps in that direction. You know, for example, there are steps, I would say really baby steps. For example, people feedback information back to the subject, and they can in fact, if you like, manipulate their activity of the brain to accomplish a particular task based on that feedback. And if you like, that's kind of a interface. But of course, you can go to, if you like, much more interesting interactions and interfaces. One can think about, for example, activating various areas of the brain or recording from various areas of the brain and use those recordings to perform particular tasks. So there are some activities that go on in there. There is one particular limitation with fMRI, however, in that respect. And I mentioned and I focused a lot on the spatial resolution, but there's also, of course, in the brain, a very important aspect of brain function is temporal resolution. And fMRI doesn't have that millisecond temporal resolution, and that becomes a bit of a limitation for doing maybe fMRI-based human-computer interfaces.
I see. Yeah, it's still feels a long ways, but it's very interesting area, of course.
Yeah.
Especially for the disabled who cannot communicate. That will be some fantastic advance, I think. But
yeah, I agree. Absolutely, I agree.
Yeah, thank you.
Yeah.
I'm so sorry, Hocam.
Yeah, no problem.
Sorry. We prepared some questions for you. Also, you mentioned some of them. Our first question is, what advice do you have for students and young researchers who are interested in pursuing a career in neuroscience?
I mean, neuroscience is a blossoming field, and there's a lot of interest, of course, in neuroscience. It's going to have huge applications in many places. Of course, not only just try to understand the brain, but medical impact, and even, if you like, in economics, marketing, you name it. I mean, neuroscience is extremely important, and so it looks like a wonderful career to pursue. And of course, based on the lecture that I was in my lecture, I would try to tell you, try to find an extremely rich environment where you can develop your education and pursue that career.
Yeah, thank you. And our second question is, how do you stay motivated and inspired in your work?
I don't know. Maybe it is sometimes I get asked that question quite a bit. Sometimes I joke about it, say, well, you have to eat your broccoli and your blueberries, but it is an internal process. And for me, what is the act of discovery and the act of bringing new knowledge is extremely exciting. Why is it so exciting for me? I really don't know. And I guess all scientists, I'm sure, are motivated by this. And if personally you have that feeling, you actually think that learning something new and discovering something new is so fantastically exciting for you that you think that life is worth living, then you are a scientist and you should pursue a career. As to why that is the case, I don't really know. But one thing that is clear, it's in fact, again, I come back to this, what I was mentioning, being in a very intellectually rich environment is so important. And I was actually earlier was talking about it. During COVID, we were all isolated, and I actually started not liking that very much. And we were not even allowed to go into our labs. And then the university started allowing us to come back. And I encourage my colleagues to come back, because to be in an environment where people are excited, they are working on new things, and they discover new things, and you see them enthusiastic, shining with new ideas, etc. And that personal interaction, just when you're having coffee or when you're having lunch. So this environment, such an environment is so fantastic. So now I try to come to the lab every day, and I come to the lab every day, and it keeps me, in fact, really motivated and inspired as well to interact with my colleagues and friends.
We can say that it comes inside of your heart.
Yes.
We can. Okay. Also, you mentioned our third and fourth question. If you want, we can pass.
Yeah, we can pass it. I think you had asked me those questions, so my lecture was kind of centered around those questions.
Okay. Our last question is, what do you consider to be the most pressing challenges facing the field of neuroscience today? How can we overcome these challenges?
Well, in neuroscience, or trying to understand the brain, there are enormous challenges. We are only just scratching the surface, if you like. I mean, we have learned quite a lot about the brain, and we can even think about potentially, even imagine that we have some vague understanding of some of how these processes work. And so there are a lot of things to tackle on the brain. I mean, if you think about it, brain function, I talked to you about whole brain activity, which is brain-wide networks that are active, then we go into mesoscopic scale, and then we go into cells, and then you go into synapses, and you have all these molecular processes, all these genetic processes that, you know, you go from single molecules to the whole brain-wide network, and it's an amazing system that has to come together to generate this incredible capabilities. So there are a lot of things that are challenging. I mean, if you just take we can talk about networks that are involved in, for example, brain dysfunction. We think that the brain dysfunction in neuropsychiatric disease are circuit problems. So you can think about those circuit problems. And again, we need better tools to map those circuit problems. But then at the same time, we have to think about problems at the level of molecules and genes and synapses, etc. So there are, it's very difficult to identify a single challenge. There are many challenges. And then there are many people working on this puzzle at different levels. And at some point, we have to bring that puzzle together. We are very far from that. How can we overcome these challenges? Really, I want to come back of course, not only we have to have, smart people working on these problems, but we have to have tools. And Brain Initiative, you mentioned the Brain Initiative. This is a very big, major effort in the United States to increase the amount of research that we are doing on the brain. Brain Initiative specifically focuses on tool development, and so it's a very important part of solving these challenges.
Okay, thank you, Hocam. Our questions is finished.
Okay.
Also, we can take more question from our participants.
I think we have no other question.
All right.
Yes.
Thank you.
Thank you for coming, Hocam.
My pleasure, and I wish you a good evening, and I continue with my day with
Have a
other
great
work
day.
still before I have a good evening. Bye,
Thank you.
everybody. Bye.
Thanks.
Have a good one.